Hello everyone, and welcome to today's The Scientist webinar. I'm Betsy Young, the technical editor for The Scientist, and I'll be moderating our discussion. Today, our panel of experts will discuss some of the mechanisms by which epigenetic modifications are known to be involved in cancer. We like our webinars to be interactive. We encourage you to send us your questions or comments at any point during the webinar, and the panelists will address these during the Q&A session following the presentations. To ask a question, simply type your query into the question box located on the bottom left-hand side of your screen. We will try to address as many of these questions as we can during our Q&A sessions. The webinar platform is user-friendly. You can move or resize any of the windows by simply grabbing them at the top or stretching them at the bottom right-hand corner. You may need to move or minimize some of the windows to see the live view. The webinar will be archived on the Scientist website, and we'll send you a link via email within a couple of days. Please note that you will not be able to download the presentation slides. I'd like to take this opportunity to thank our webinar sponsors. Rockland Immunochemicals, Inc. offers each academic, biopharma, and diagnostic professional thousands of antibodies with the aim of providing the right antibody that is the perfect fit for every occasion. With facilities in Pennsylvania for over 50 years, Rockland manufactures products ideally suited for integration into critical assays such as Western blotting, immunohistochemistry, immunofluorescence microscopy, ELISA, and flow cytometry. No matter the context, basic research to disease therapy, phosphorylated to methylated and beyond, Rockland embraces the challenge to design, produce, validate, and deliver the best antibodies and life science reagents available in the market today. With Rockland, your solution is complete. To learn more, visit rockland-inc.com. Swift Biosciences is transforming the next-generation sequencing space by providing new technologies for sample and library prep methods. Swift is the only company to have products for all three major next-generation sequencing platforms. Based on its own unique and patented technologies, Swift allows researchers to use lower input amounts, including heavily damaged FFPE samples. Capable of generating libraries from as little as 1 nanogram of input, Swift is continually improving its technologies with a focus on single-cell experiments. Currently, Swift Biosciences has a broad product portfolio supporting multiple next-generation sequencing applications for personalized medicine. These products include the Excel NGS-2S DNA Library Kit, which delivers unprecedented performance when working with low-input samples like circulating cell-free DNA and circulating tumor cells, and Excel amplicon panels for targeted sequencing. Individuals can select from a predetermined 56G oncology panel or design a custom panel for their genes of interest. Visit swiftbiosci.com to discover more. Enzo Life Sciences is a manufacturer of labeling and detection technologies from genome to whole cell. Enzo's products are backed by innovative technology platforms and a deep patent portfolio. With 40 years' experience, Enzo provides a complete toolbox for screening epigenetic regulators ranging from extraction kits, enzymes, modification activity and quantification assays, and small molecules. Enzo offers non-radioactive HDAC and sirtuin screening assays in chemiluminescent, fluorescent, and colorimetric formats. Their high-quality assays utilize patented substrate developer chemistry in combination with high-activity, high-purity enzymes to deliver more high-quality hits. Visit enzolifesciences.com for more information. Our sponsors have provided us with some helpful resources related to epigenetics and cancer research, and we've posted these in our resource list located on the left-hand side of the screen. You can access and download these documents at any time during the webinar. And with that, let me introduce the first speaker, Dr. Ahmad Khalil. Dr. Khalil is currently serving as an assistant professor of genetics and genome sciences at Case Western Reserve University School of Medicine in Cleveland, Ohio. He received his PhD in 2005 from the University of Florida College of Medicine. Dr. Khalil completed his postdoctoral training in 2010 at Harvard Medical School and the Broad Institute, where he played a major role in discovering human long non-coding RNAs. More importantly, he was the first to demonstrate that numerous lncRNAs interact with histone-modifying complexes to regulate the epigenome and gene expression. At the end of 2010, Dr. Khalil started his own lab, and his work focused on elucidating the roles of lncRNAs and epigenetics in cancer biology. His lab recently made a seminal discovery that dysregulation of lncRNAs could be a major mechanism driving aberrant epigenomes of cancer cells. He's received several awards for his research, spanning his graduate school career to present. Dr. Khalil?
Thank you, Betsy. Good afternoon, everyone, and thank you for joining us. I would like to take this opportunity to thank Biosciences for organizing this wonderful seminar. And I'll start with my very first slide. So I would like to just remind you that will be discussing mechanisms of epigenetic alterations in cancer. I'll talk mostly about DNA methylation today in the context of colon cancer. So it has become very clear to us and many others that human cancers are driven by both genetic and epigenetic alterations. And just a quick reminder that epigenetic is the biological information that is not encoded in our DNA sequence itself, but this information is critical for proper gene expression, especially in the context of tissue-specific expression. So some of the major epigenetic factors that we have studied for many years are DNA methylation and histone modifications. And we and others have discovered that the epigenome of cancer cells have significant changes in these epigenetic modifications, DNA methylation and histone modifications, when you compare cancer cells to match normal tissues. So these observations have suggested that the changes in the epigenome could be contributing to the initiation, progression, and the aggressiveness of different cancer types. What is not known is what are the mechanisms that drive these epigenetic alterations during tumorigenesis. So simple explanations such as mutations in the genes that are involved in epigenetic regulations have not been fruitful because those are actually very rare in many cancers. So the simple explanations that most of us thought of at the beginning did not turn out to be completely correct. In some cases they are, but not the full story. So like I said, I want to focus today on one of these epigenetic modifications just to really get into depth talking about these alterations and the potential mechanisms. So I'm going to focus on DNA methylation, alterations in cancer. And there's two types of changes in DNA methylation in cancer. The first one is gain of DNA methylation or hypermethylation at promoters of tumor suppressor genes. There is another type of change in DNA methylation. It's hypomethylation, which is the loss of genome-wide DNA methylation, which occurs at both promoters of oncogenes as well as many intergenic regions of the genome. While the gain of methylations at the promoters of tumor suppressors is easy to explain because they lead to the repression of tumor suppressors, the loss of methylation, especially in the intergenic regions, has been less clear what the biological significance of this, although we have some insights that I will discuss today. The major question is, what are the underlying mechanisms that are driving genome-wide DNA hypomethylation during tumorigenesis? And I would like to share some data with you today that suggests that there is a role of these lncRNAs in driving this hypomethylation. So just a very quick reminder that DNA methylation takes place on cytosine residues. There's a number of enzymes, which I will discuss in the next slide, that catalyze this modification to convert it to 5-methylcytosine. And these enzymes in mammalian cells are known as DNMT1, DNMT3A, and 3B. DNMT2 initially was thought to be a DNA methyltransferase, but it turned out to be an RNA methyltransferase. And DNMT3L lacks the enzymatic activity of the other methyltransferases. Although in general we tend to think of DNMT1 as the maintenance DNA methyltransferase and 3A and 3B as the de novo methyltransferases, it turns out, like everything in biology, it's a little bit more complex, where sometimes DNMT1 can also function as a de novo methyltransferase. So we still have a lot to learn about these enzymes and how they regulate methylation. So specifically today, I'm going to focus on colon cancer. It was discovered back in 1985 that one of the major alterations that occurs very early in the process of colon tumorigenesis is the loss of DNA methylation. So as you can see in this diagram, which was adapted from Bert Vogelstein, who is one of the leaders in colon cancer, the first hit in colon tumorigenesis is a mutation in a gene known as APC. But the second hit is actually not a genetic mutation, it's an epigenetic alteration, which is DNA hypomethylation, which leads to the progression toward adenomas. And then you have KRAS activation and loss P53 to reach the carcinoma stage. And this observation has now been confirmed by many other independent laboratories. However, the mechanism that leads to DNA hypomethylation in the early stages of tumorigenesis is not known. And that's one of the things we focused on to try to figure out the potential mechanism. So again, quickly, DNA hypomethylation in colon cancer was first reported back in 1985 and confirmed by many subsequent studies. As I mentioned briefly earlier, initially it was thought that maybe this is due to mutations in DNA methyltransferases, but it turned out that actually these mutations are actually rare in colon cancer. And the other paradox that actually these DNA methyltransferases levels are increased in colon cancer and other cancer types. So again, it's not due to repression of DNMTs as one could hypothesize. So we wanted to test a very novel and exceptional novel hypothesis that maybe the changes in DNA methylation patterns are impacted by changes in lncRNAs that regulate these DNMTs, which leads to changes in genome-wide DNA methylation. So when we first proposed this hypothesis, we didn't even know if DNMT1 or other DNA methyltransferases interact with regulatory RNAs like lncRNAs. So the very first step was for us to try to figure out if there is an interaction, which I'll show you the data in the next few slides. But since I'm spending quite a bit of time talking about these lncRNAs, I would like to just kind of give you a brief overview of what they are and what we know about them. So in 2009, while I was a postdoc in the RIN lab, we published a paper where we found that the human genome encodes thousands of long intergenic non-coding RNAs. So these are regulatory RNAs that are transcribed far away from protein-coding genes. They don't have any protein coding capacity, but they play many important biological roles. The other major discovery we made that these lncRNAs many of these lncRNAs interact with chromatin-modifying complexes, such as the polycomb repressive complex 2, and via these interactions, they actually can regulate gene expression, in some cases at the transcriptional level, by modulating epigenetic modifications. So these observations suggested to us that if you alter the expression of lncRNAs, you can potentially alter the epigenetic landscape of cancer cells. So based on these studies, we proposed the following model. So these lncRNAs are transcribed by RNA polymerase II, and then once they are transcribed and spliced, they can form some form of secondary structure that allows them to recognize different protein complexes. We are using here PRC2 as an example. And this protein-RNA interaction allows these complexes like PRC2, which do not bind specifically to specific DNA sequences, to be targeted to specific parts of the genome where they can now change the histone modification patterns, or in the case of DNA methyltransferases, potentially DNA methylation patterns. So again, disruption of some of these lncRNAs could affect the way these PRC2 or other complexes are targeted to the genome. So let's now go back to this idea of DNA hypomethylation in colon cancer. So we wanted to test, again, this hypothesis that DNMT1 interacting lncRNAs lead to this DNA hypomethylation. So first, we had to test if DNMT1 interacts with lncRNAs. And to do this, we performed an experiment known as RIP-seq, which stands for RNA co-immunoprecipitation, followed by RNA sequencing. So the way this technology works, you IP your protein of interest, in this case DNMT1, and use cell lysates that have been cross-linked. You isolate the associated RNAs that co-IP with your protein of interest, and then you perform next-generation RNA sequencing and bioinformatic analyses to identify RNAs that interact with that specific protein. So the first part of the experiment was to confirm that we actually specifically pulled down DNMT1. So as you can see in this Western blot, when we use a flag tag DNMT1 cell line, we can use the flag antibody to IP DNMT1 and confirm by Western that we are specifically IPing DNMT1, but not in our IgG negative control. We used two antibodies for two highly abundant nuclear proteins to show that we are specifically IPing DNMT1 without much background, and we used U170K and histone H3 antibodies to show that. So once we confirm that we can specifically IP DNMT1, we sequence the RNA that comes down with our IP, and we use stranded RNA sequencing. And then we identified, or we asked which RNAs, whether it's mRNAs or lncRNAs, that are showing enrichment in our IP versus input. So this is the heat map showing the three biological replicates of our DNMT1 RIP-seq as compared to the input sample. As you can show, there is on the top, shown in red, there is potentially some RNAs that specifically associate with DNMT1 at much higher enrichment than our input sample. So using this analysis, we identified approximately 148 lncRNAs that potentially interact with DNMT1. And the highest fold change for the strongest lncRNA that interact with DNMT1 was about 41-fold above input. Since this is RNA-seq, we can also ask, are there mRNAs that also associate with DNMT1? And we found very few mRNAs to associate with DNMT1. The highest fold change was also about 7-fold as compared to the 41-fold of the lncRNAs. So just to put the two results side by side, so we found many more lncRNAs that interact with DNMT1 versus very few mRNAs. Actually, those 31 mRNAs, when we looked closely at them, many of them are predicted open reading frames, and some of them might turn out also to be non-coding. Just a quick reminder that mRNAs are expressed at much higher levels than lncRNAs in all cell types. So this suggests that this IP is not specifically identifying highly abundant RNAs. If that was the case, you would see the opposite result in this case. So another control that we did was to ask another simple question bioinformatically. Are the lncRNAs that we are observing to be co-IP'd with DNMT1, are they expressed at much higher levels than other lncRNAs, because that would suggest non-specific interactions. So if you look at these graphs, you would see that the lncRNAs that interact with DNMT1 are not expressed at higher levels than the lncRNAs that do not bind DNMT1. Again, this also provides another evidence for specific interactions rather than non-specific interactions. So now we have 148 lncRNAs that interact with DNMT1, and we wanted to identify top candidates to start doing functional studies on these lncRNAs and ask their role in regulating DNA methylation. So to narrow down the list, the first thing we did, we intersected the lncRNAs that interact with DNMT1 with lncRNAs that are dysregulated in colon tumors versus matched normal tissues, and we obtained that data set from the Cancer Genome Atlas. So this allowed us to narrow down the number of lncRNA candidates to about 20 or so. Then we took the top lncRNAs and we examined the expression of them in a panel of normal human tissues. And this allowed us to identify lncRNAs that show high tissue-specific expression in the colon. And here is a graph of one of our top candidates, which we now refer to as DACOR1. And this is its expression in normal human tissues. And as you can see, it has the highest expression in the normal colon and followed by the small intestine. If we look in by RNA in situ hybridization of the expression of DACOR1, we found it to be expressed in cells known as the colon crypts, and these are the cell types that are actually give rise to colon cancer. So the arrows are pointing to the lncRNA expression in this tissue section of a normal colon, and the smaller panel is showing that DACOR1 is expressed and retained in the nucleus. So this is consistent with its interaction with a nuclear protein like DNMT1. So we wanted to confirm that DACOR1 is actually dysregulated in colon cancer based on our analysis in the Cancer Genome Atlas data, or the TCGA data set. So we obtained RNA from both normal colon samples and from 21 patient-derived colon cancer cells in collaboration with Dr. Markowitz here at Case Western. And as you can see that in blue is the normal colon, red is the colon cancer samples, and you can see that DACOR1 is suppressed in many of these patient-derived colon cancer cell lines. So this confirmed our observation in the TCGA dataset. So just to quickly remind you what we know about this lncRNA, we know it interacts with DNMT1. It's highly expressed in normal colon tissues. It's downregulated in colon cancer. So now we have two additional key questions. The first one is, does DACOR1 affect DNA methylation based on its interaction with DNMT1? And the second question, does DACOR1 have a role in colon tumorigenesis? So the first thing we did is to confirm the interaction between DACOR1 and DNMT1 by independent RIP experiments, but now by qPCR instead of RNA sequencing. As you can see in the left panel that DACOR1 interacts with DNMT1 and enrich over an IgG control. We do the same analysis for a highly abundant nuclear RNA such as U1, and you can see there is no enrichment of this highly abundant nuclear RNA with DNMT1 versus IgG. So this gives us additional confidence that there's a specific interaction between DACOR1 and DNMT1. Then we asked, what happens to these colon cancer cell lines if we were to re-express DACOR1 in the cells that represses its expression? So we took two patient-derived colon cancer cell lines labeled as V481 and V852 at the top. And in three independent biological experiments, we asked what happened to the methylation patterns when we induce the expression of DACOR1. So in these heat maps, the blue is low methylation, the red is high methylation. And in both cell lines, what we noticed in all three replicates, that when we induce the expression of DACOR1, we start to see enhanced DNA methylation at specific sites in the genome. This analysis was performed using Illumina 450K arrays, which only cover about 2% of all CPG sites in the human genome. So next, we wanted to ask if we would observe a stronger effect if we use the technology that allows us to assess many more CPG sites in the genome. So to do this, we switched to using reduced representation bisulfite sequencing assay, So this is dependent on DNA sequencing and allowed us to assess the changes in DNA methylation at several million sites in the human genome. Again, it's not a complete coverage of all 28 million sites in the human genome. That would require whole genome sequencing, but this assay covers several millions of those CpG sites. So what you are looking at in this histogram is on the y-axis is the number of CpG sites, and on the x-axis is changes in methylation. So this, at 0, there is no change in methylation between cells that were treated with a control versus our lncRNA DACOR1. And each bar graph represents 5% in change in methylation. So as you can see that the majority of changes in methylation we are observing was increased DNA methylation. If, for example, you use a 0.25 or 25% difference in methylation between the two conditions, we identify over 100,000 CpG sites that become methylated in response to the re-expression of DACOR1. Again, this data supports our previous observations that DACOR1 enhances DNA methylation, potentially by regulating DNMT1 activity. So I'll show you the distribution of those CpG sites per chromosome. So on the x-axis is each one of the human chromosomes, and on the y-axis is the number of CpG sites. This data is only for CpG sites that have a difference in methylation of 30% or more between the two conditions. So this is not all the changes from the previous slide. And as you can see, the effect on DNA methylation is observed virtually on all human chromosomes. So this data suggests that DACOR1 really impacts genome-wide DNA methylation. So next we wanted to assess if the induction of DACOR1 actually impacts colon cancer cells. So we performed colony formation assays in 3 different patient-derived colon cancer cell lines labeled at the top with V followed by a number, V481, V852, and V866. And as you can see from the quantification at the bottom, that when we induce the expression of DACOR1, this leads to reduced colonogenicity of these colon cancer cells in all 3 independent lines. So this suggests that DACOR1, in addition to impacting genome-wide methylation, it could also be working as a tumor suppressor lncRNA. So we wanted to understand how is DACOR1 impacting these colon cancer cells. So to do this, we performed RNA sequencing of colon cancer cells with either a control or DACOR1 infected via lentivirus. And then we performed differential gene expression and pathway analysis following these RNA-seq analyses. And we identified a subset of DACOR1 regulated genes that are also affected in colon tumors based on the analysis from colon tumors versus matched normal tissues in the TCGA dataset. And some of these key pathways that emerged included the TGF-beta pathway, which is known to be involved in colon cancer, and several cellular metabolism pathways that also have a role in tumorigenesis. So here's some validation of some of these genes that are involved in TGF-beta. So those genes that are impacted by DACOR1 are known to inhibit the repressors of the TGF-beta pathway. So this suggests that by re-expression of DACOR1, you are inhibiting the inhibitors of this tumor suppressor pathway. So for example, SMAD6, a gene that is known to be highly expressed in colon tumors. So as you can see in normal tissue in blue versus colon tumors in red, SMAD6 expression is much higher in many of these colon tumor samples. So DACOR1 is repressing SMAD6, potentially reactivating some aspects of the TGF-beta pathway. The other set of genes that were really interesting as well is genes that involved in metabolism. So we found that DACOR1 represses genes that are known to be highly expressed in colon tumors, including genes involved in serine metabolism, for example, PHGDH. So let me show you the data for this gene. So PHGDH is highly upregulated in colon tumors, as you can see again here in red versus blue, and it's known to be required for serine biosynthesis. So serine biosynthesis is a really important aspect of tumorigenesis. So if you look at this timeline in a review article, so in 1955, it was discovered that de novo serine biosynthesis was first observed in tumors. And then about almost 4 decades later or more, in 2011, it was shown that actually de novo serine and glycine metabolism is necessary and sufficient for cell transformation in malignancy. So this is a very important aspect of the tumorigenesis process. And DACOR1 seems to be regulating the genes that are involved in these metabolism pathways. So here's the confirmation of our RNA-seq data at the protein level. You see that when we induce the expression of DACOR1, we are repressing PHGDH level at the protein levels. And serine levels, which are regulated by PHGDH, actually important for the enzymatic activity of another protein called PKM2. So PKM2 is a very important gene in cancer metabolism. We could spend an entire hour talking about this gene. Obviously, we don't have the time to today. But what we found that when we express DACOR1 and repress PHGDH, we can reduce the activity of PKM2 as shown in the graph on the right-hand side. So this is a very significant finding, again, because PKM2 really plays a really important role in cancer metabolism. Finally, another gene that was impacted by DACOR1 induction is a gene known as CBS, which indirectly regulates S-adenosylmethionine levels, which are important for DNA methylation. So it's well known that SAM is required for DNA methylation by DNMTs, that increased SAM levels could contribute to the enhanced DNA methylation that we are observing. So we measured SAM levels as shown in the graph, and we showed that when DACOR1 is induced, we have increase in SAM levels in these cells. And again, this is really important because SAM is sufficient to inhibit tumor growth as shown by previous studies. So again, this lncRNA that interacts with DNMT1 could be doing two different things. One is regulating DNA methylation by direct interaction with DNMT1 and chromatin, but also by regulating genes that regulate cellular metabolism levels such as S-adenosylmethionine that is really important for DNA methylation. So to test that DACOR1 is doing both mechanisms, we asked by a technology known as a ChIRP-seq if DACOR1 interacts with chromatin. So as you can see in this graph that we confirmed that we are specifically pulling DACOR1 RNA from our cross-linked cell lysates, and then we map the genomic occupancy sites by DNA sequencing of DACOR1. And what we found that actually DACOR1 binds to many sites in the genome. So if we have a very stringent cutoff of 20-fold enrichment, we identified over 338 sites. If we reduce our stringency to 10-fold enrichment over the control probes, then we identify over 2,000 sites in the genome. If we take the high stringency cutoff and we just ask for those sites that are near protein coding genes, so it's about half of those 338 sites, how many of them overlap with known differentially methylated regions in colon cancer, we find a significant overlap, and the p-value is shown in the graph. So again, this data suggests that DACOR1, in addition to interacting with DNMT1 and localizing to the nucleus, it also directly interacts with chromatin at specific sites. So based on these observations, we propose the following model. that in colon cancer cells, DACOR1 become repressed, and the repression of DACOR1 leads to the upregulation of PHGDH and PSAT1, which are two genes involved or essential for serine biosynthesis. So this leads to an increase in serine biosynthesis and increased PKM2 activity, which is essential for cancer cells metabolism. And the increase in CBS and potentially the decrease in SAM levels reduces the levels of genomic DNA methylation and leads to genomic instability, which is a hallmark of cancer cells. So again, this is a proposed model. There are probably other aspects of how DACOR1 is impacting the genome, but these are some of the key pathways we have identified so far. So just to quickly summarize what I showed you today, we identified that DNMT1, which is the major DNA methyltransferase, interacts with specific lncRNAs in colon cells, that DNMT1 interacting lncRNAs orchestrate genome-wide DNA methylation in human cells. We think, based on the both the phenotype and the gene expression analysis, that DACOR1 could be a novel tumor suppressor lncRNA. It become repressed in colon cancer, but the induction of this lncRNA seems to be sufficient to restore genome-wide DNA methylation at thousands of sites throughout the genome. So we believe that these findings provide much-needed mechanistic insights into DNA hypomethylation that have been observed during colon tumorigenesis. If you have additional questions about this work, you can read our article that was published at the end of 2015 in Human Molecular Genetics, and we published some of these findings in that paper. Finally, I would like to end by acknowledging all the people that have contributed to this work. This work has been done by very talented students in the lab that were able to work these difficult technologies and ask these important questions. Our collaborators that have provided some of these key cell lines and reagents to be able to do these experiments. And again, I would like to also thank our funding sources. But again, I would like to thank the scientists for this great opportunity to present this work. Thank you very much.
Thank you, Dr. Khalil. As a reminder to our audience, if you have any questions for the panelists, you can type them into the question box at any time. The panelists will answer them during the Q&A session. Our next speaker is Dr. Lanlan Shen. Dr. Shen is an associate professor at the USDA ARS Children's Nutrition Research Center at Baylor College of Medicine in Houston, Texas. She received her MD in 1992 from Jiangsu University Medical School in China and received her PhD in 1997 from Shanghai Jiaotong University Medical School. Dr. Shen's long-standing research interest has been cancer epigenetics, with an extensive publication record dating back to 2002, when she was a postdoctoral fellow in Dr. Jean-Pierre Eis's lab. Currently, the research in her laboratory is focused on understanding how epigenetic mechanisms drive normal cellular differentiation and how epigenetic dysregulation drives cell proliferation and tumor formation. Dr. Shen's group has substantial experience in epigenetics and cancer biology, including epigenomic profiling, molecular biology, and mouse models. Dr. Shen?
Oh, thank you. Good morning. Good afternoon, and thank you for inviting me to this event. Today, I'm going to talk about our recent study to determine the functional consequences of aberrant DNA methylation in cancer. Over the past decades, it has become clear that cancer is not only a genetic disease, but also an epigenetic disease. Epigenetic silencing, particularly by alterations in DNA methylation, is frequently found in human cancers. Indeed, it was proposed almost more than 25 years ago by Robin Holliday that such epimutation can act as one of nuisance to heads to drive tumorigenesis. So this is one example by a DNA repair gene Oh, okay, this is, let me go back. So in fact, I need to take back just to give a little bit introduction of how promoter methylation regulate transcription, because in our genome, more than half of all gene promoters contain a CpG-rich region called CpG islands or CGI, and methylation at this CGIs is often correlated with stable gene silencing. So this is actually one example by a gene called MGMT. This is a DNA repair gene, and it is commonly methylated in patients with colorectal cancer. In fact, almost half of the patients shows the methylation of MGMT gene. So showing here is the immunohistochemical staining of MGMT expression in tumors with or without MGMT methylation. So on the left is a methylated tumor showing no expression in the cancer cells. In contrast, you can see a nice positive staining of MGMT in the adjacent normal cells. On the right is an unmethylated tumor, and the MGMT expression is highly expressed. In addition, the importance of DNA methylation in cancer has been strongly supported by clinical studies. For example, we have analyzed DNA methylation of multiple promoter CGIs in patients with myeloid dysplastic syndromes. And this patient, they were from 3 independent cohorts, as shown here, represented by 3 different panels, the figures you can see here. So in each panel, each of the column indicates a patient, and each row indicates a CGI we analyzed. The green indicates low level of methylation, and red indicates high methylation. What we found is that for all the 3 cohorts we analyzed, the patients with a high level of methylation, they all show significantly poor prognosis. So clearly, the increase of methylation is correlated with the clear clinical outcomes in the patient. This has been shown not only in the myelodysplastic syndrome patients, but also shown in many other cancer types. On the other hand, we also know that cancer is a complex disease which involves combined genetic and also epigenetic changes. In colon cancer, we actually show that some subset groups of patients with high degree of aberrant methylation, which is a phenomenon known as CpG island methylated phenotype, or SNP, are associated with distinct genetic mutation patterns. For example, the SNP1 group is closely associated with BRAF mutation and also microsatellite instability, whereas the SNP2 group is strongly associated with KRAS mutation, and the SNP negative group has a high rate of p53 mutation. So clearly, these association studies have provided significant insights on the role of DNA methylation in cancer. They have also raised a long-standing question in the field, and that is whether aberrant DNA methylation is a cause or a consequence of cancer. Interestingly, many cancer-related promoter hypermethylation, you can actually detect them in the normal aging tissues. For example, this scatter plots showing here is the correlation between methylation level of an indicated gene versus age in the normal appearing colon mucosa samples. What you can see is that there is a general increase in the percent methylation with age for all 6 genes we have analyzed, suggesting that aberrant DNA methylation could predispose to the malignancy. Directly Testing this hypothesis, however, requires an experimental approach to induce local-specific promoter CGI methylation, ideally in vivo, and can demonstrate the functional role of those aberrant DNA methylation. Towards this end, we have been pursuing an epigenetic engineering approach by utilizing cis-acting DNA sequence that can regulate the methylation of nearby genes. And we define it as pro-methylation cis element. So the idea was built upon our discovery that specific cis-acting DNA motifs are associated with a class of normally methylated and silenced promoter CGIs. Using stable transgene experiments, we show that an engineered just 140 base pair cis element, in fact, could induce de novo methylation throughout the promoter CGIs of the transgene. So this positive data prompted us to generate a mouse model with targeted knock-in of the cis element at the P16 locus. We choose P16 as a model gene because P16 promoter CGI methylation is among the most common and earliest epigenetic events in human cancer. So the gene product of P16 regulates the ability of retinoblastoma protein to control the G1 to S transition of the cell cycle. And the tumor suppressor function of P16 has been convincingly demonstrated by studies of P16 knockout mice, and the animals are predisposed to either spontaneous tumor or carcinogen-induced tumor development. But the question here is, how about P16 methylation alone can itself induce tumorigenesis? Why we ask this, I wanted to point out here, is that when you compare the genetic and epigenetic alterations of P16 in human cancers, you often find that P16 inactivation is more frequently associated with promoter hypermethylation than genetic mutations. For instance, you can see in sarcoma, liver, or colon cancers, you often find promoter methylation with very rarely genetic deletion or mutations. So we decided to introduce the cis element 1 kb upstream of the P16 transcription start site in mouse embryonic stem cells through a standard homolog recombination. This insertion was designed to leave the core promoter of P16 intact, and also we try to avoid affecting the neighboring genes, which are located 11 kb away, as indicated here by the gene structure of the P16 locus. As a negative control for the insertion effects, we also knock in a control element at the same region. So we first studied the changes of DNA methylation in the knock-in mouse ES cells before and after induced differentiation. And here, each panel is a methylation profiling at the CPG sites, spanning the knock-in sequence and also the endogenous P16 promoter. So the left panel shows the control knock-in ES cells, and the right panel shows the cis element knock-in ES cells. What we found is before differentiation in both ES cells, most CPG sites are either unmethylated or show low levels of methylation. Upon induced differentiation in the control ES cells, a few CPG sites within the knock-in sequence, we actually see some modest increased DNA methylation levels, but the methylation remains low within the endogenous P16 promoter regions. In contrast, on the right, what you can see is in the cis element knock-in ES cells, differentiation induced dramatically increased methylation both at the cis element and also at the endogenous P16 promoter. When we analyze the gene expression of P16, what we found is this induced promoter methylation during differentiation leads to strong transcription repression. Importantly, when we treat the cells with a hypomethylating agent, we were able to reactivate the P16 expression in the cis-knocking differentiated ES cells. This provides further validation that P16 silencing is not simply due to, for instance, some side effects of cis element knocking. In addition, we also observed significantly increased repressive histone modifications, for instance, the K9 dimethylation, and also the modifications of K27 trimethylation. And this suggests that the cis element mediated epigenetic silencing could be reinforced by changes in chromatin configuration. And more recently, in order to provide a mechanism of action for how the cis element mediates targeted methylation, we have used DNA pull-down in combination with mass spectrometry analysis to screen for proteins that bind to the cis element. And this unbiased approach has allowed us to discover and validate that DMT3L and 3A, they are recruited to the cis element and contribute to the induced de novo methylation. I just want to remind you that these two enzymes are known to be show the de novo methylation activities. We have also analyzed the cis element-induced P16 methylation in mice in vivo, and I need to mention that these mice, they were homozygous for the cis knocking alleles, and we have also removed the drug selection cassette. So the idea is to restore the P16 locus to as close to as undisturbed as possible. So only is this 140 base pair cis element and few of the LOXP sites and the FRT remain sites. When we analyzed the mice at birth, we found that in all somatic tissues, the initial methylation was within the cis element. With aging, we saw this methylation starts to spread towards endogenous promoter. And this methylation spreading appears to be tissue-specific, and we found that was most obvious or significant in highly proliferative tissues, including spleen and intestine. As expected, we also found significantly reduced transcription in the methylated tissues. You might notice that the level of increased methylation appears to be modest. One of the potential explanations is that we analyzed the level of increased methylation in the whole tissues which contain a mixture of different cell types, and the methylation spreading could also be, in addition to tissue-specific, but also cell type-specific. So to address this possibility, we used mouse embryonic fibroblasts, or MEF, as a homogeneous cell population, and also as a classic model for cell senescence. So here, what we are monitoring is the methylation change during passaging of the MEF generated from either cis knocking or control knocking animals. So what we've also in this graph, the passage numbers are indicated next to the time points we collected, and also the days of the cells under the culture is indicated by the x-axis. What we found is that for the control knocking MEF, we were only able to passage them for about 8 passages, and the cells become senesced. And consistent with the mouse ES cell studies, we found there's little changes of P16 promoter methylation, and the levels remains very low in those cells. In contrast, the cis knocking mouse bypass the senescence and become immotile spontaneously. And consistent with our hypothesis, the cis element induced progressive and extensive P16 promoter methylation. When we then compare the transcriptional changes for this mouse under the passaging side by side, and it becomes clear that the methylation increases precedes the transcriptional silencing of the P16 gene. So putting all these results together, we conclude that the knocking cis element successfully induced the targeted P16 epimutation. So now the obvious question is whether the induced P16 hypermethylation is tumorigenic in mice. And the answer is yes. We found significantly increased incidence of spontaneous tumor development in the mice carry the cis knocking alleles. And those tumors we identify include sarcoma, lymphoma, and also lung adenocarcinoma. And we can also confirm that the P16 promoter is indeed methylated in the bulk of the tumors we have collected and analyzed. Furthermore, by the survival analysis, we show that mice carrying engineered P16 epimutation had significantly shortened tumor-free survival. And consistent with Knutson's two-hit hypothesis, if the animal carried one allele with germline deletion and the second allele with engineered P16 epimutation, that had accelerated tumor development and also significantly quicker the tumor-free, the early death of the animals. So in summary, I hope I have convinced you today that our studies provide direct evidence for a driving role of P16 epigenetic silencing by promoter methylation in tumor formation and progression. Our straightforward epigenetic engineering approach should be broadly applicable for future functional epigenetic studies, not only in cancer studies, but also for other epigenetic-related diseases. In addition, the model systems we have developed will be valuable in the ongoing search for the new and improved epigenetic therapy and prevention. And with this, I would like to acknowledge the people who contribute significantly to this work. Dahai Yu did most of the studies I presented here today. I also acknowledge the contributions of my colleagues and collaborators at the Children's Nutrition Research Center, Baylor College of Medicine, and Texas Children's Hospital, including Rob Waterland, Grant Guang, Ashley Chen, Pumin Zhang, and my close collaborators, Yi Wang, Jing Chen, and Deborah Shady. Our work was supported by grants from Sidney Kimmel Foundation and USDA. And thank you.
Thank you, Dr. Shen. We've received several questions from the audience, so let's jump in. I'll get started with Dr. Khalil. How exactly did you induce DACOR1 in cells?
That's a very good question. So we used a lentivirus, and we used two different promoters. We used a CNV promoter for overexpression and PGK promoter for expression that is comparable to the physiological levels. So we used these two different systems.
Great. Do you think there's any significance, Dr. Khalil, to the lack of change in methylation on the Y chromosome when DACOR1 is expressed in colon cancer cells?
So that's a really good question, and I'm not sure if this is due to technical issues or actual biology here. So the Y chromosome is mostly repetitive elements. So remember when we are analyzing our genome-wide analysis by reduced representation by sulfide sequencing, the repetitive nature of the Y chromosome tend to complicate some of these analyses. So it could be due to some type of technical issue with mapping reads back to the Y chromosome, or it could be due to some biology that could be very exciting. We don't know yet.
Thank you. Dr. Shen, are there examples in nature of cis-acting elements that epigenetically modify nearby sequences, and are they able to be inserted using CRISPR?
That's really important question. So the cis element we found actually they frequently occur at the normally in humans, normally methylated, because several years ago we used the genome-wide studies and we were able to identify, even though it's only 5% of the human promoters, actually indeed they are methylated with the CPG island. So they do associate with normally methylated promoter methylation in the nature's situation. So what we did is we take the human sequence and knock into the mouse genomes, and the question is, can we use other next generation engineering approach to be more fast or quicker? That's actually is a goal we are trying to achieve.
Thank you. Dr. Shen, you studied P16 promoter methylation and silencing in MEFs. Were they immortalized with E1A?
No. So all the MEFs are they are generated just right from the mouse, knocking mouse. So we did not use any immortalization approach for those cells, and they are spontaneously become immortalized, and because the cis element induced the P16 promoter methylation and the silencing.
Thank you. Dr. Khalil, you presented data on the methylation status of the genes. Is it likely that other epigenetic modifications are taking place as well, and do you have any evidence to support this?
It's very possible. We obviously focused on examining DNA methylation because DACOR1 interacts with DNMT1, but it's possible that is a secondary effect of inducing DNA methylation that you start to change the chromatin modifications at those genes. We have not yet looked at those, but something we would like to look at.
Thank you. Dr. Khalil, recent evidence suggests that there are small peptides encoded by lncRNAs. Do you expect that you might find similar small peptides and that these peptides might influence DNMT1, or do you have plans to search for them?
So this is a really important question that we frequently get. So let me clarify. So the human genome encodes probably 20 to 25,000 long non-coding RNAs, and the number can change in the future, but that's the current estimate. And there was a very small number of studies that have found some of these long non-coding RNAs to encode short peptides in the range of, very few examples. And that doesn't surprise me because, to classify that many and annotate that many long non-coding RNAs, there's always some exception to the rule. But there's two things. One, you have to remember that many of these long non-coding RNAs are retained in the nucleus. So by the retention in the nucleus, they cannot be translated into protein. The ones that are cytoplasmic, some of them could potentially encode short peptides. So they could have dual roles. They could work as a regulatory RNA, but can also have protein coding capacity. This is actually true for some protein coding genes, where there is evidence that in addition to the protein coding capacity, the 3' UTR could have an independent function. So we still have a lot to learn about the transcriptome and what is translated into protein and what is not translated into protein. But specifically in terms of DACOR1, it's retained in the nucleus. It cannot be translated. So its role in DNMT1 function is dependent on the RNA itself.
Thank you, Dr. Khalil. And this one is for you as well. Does lncRNA bind to specific DNA to regulate expression?
So this is part of understanding the mechanism of lncRNAs. So using the ChIRP-seq data, we can say that this lncRNA is in close proximity or binds to this genomic region, but we cannot say that the interaction is direct. The technology doesn't allow us to answer that part of the question. And there is a debate right now in the field whether lncRNAs directly interact with DNA through triplex formation, or they interact with DNA via some type of protein complex that allows them to, that interacts with both the DNA and the lncRNA. We don't know the answer to that important mechanistic aspect of lncRNAs. One of the surprises that people have found that you would think that something like the exist RNA that silences the inactive X chromosome would recognize specific motifs on the X chromosome, but that has not been shown to be the case. So if I have to guess at this point, I would say there's probably some type of mediators between the DNA and the lncRNA, which are yet to be identified.
Thank you, Dr. Khalil. Dr. Shen, if you can reduce survival with a targeted epimutation, could you conversely increase survival with a different epimutation, and how?
So this really is what I have shown today is really specifically manipulate P16 promoter only. And this is important because by that way we can really understand how the specific promoter methylation, whether it has functional consequences. So because this proof of principle will allow us really further on to use similar approach to targeting other regions. For instance, we could targeting the genes which has no known functions, and then address the question whether they really plays a role in tumorigenesis, whether they are a driver or bystander of tumorigenesis, or whether they could even has opposite effect on the tumorigenesis. So these are the really interesting questions can be addressed in the future, but we haven't really tried to test it so far.
Thank you. Dr. Shen, what are the specific benefits of your epigenetic engineering approach over other similar approaches?
That's a very good question. So they are actually recently there are several papers published showing the epigenetic engineering specifically targeted DNA methylation at the endogenous locus. One you could potentially use, for instance, TALEN or CRISPR, and driving the de novo methyltransferases to a specific locus. And as we all know that the current next generation engineering approach, we do still have a concern of the off-target effects where the cis element is based on the traditional knock-in. So this is very specific and precise to the endogenous locus. And also what has been shown so far in the literature by the next generation sequence or next generation engineering approach, what has been shown, they are all based on the in vitro studies. How or whether this can work in vivo, it remains to be determined. What I think most importantly is to induce methylation not only at a few CPG sites, but rather as a domain. And that is something we need to be able to really induce a stable transcriptional changes. And that's really, I think, separates our approach differently from the other approaches.
Thank you, Dr. Shen. Dr. Khalil, is it reasonable to expect that DACOR1 downregulation would be beneficial in other tissue types?
That's a good question. I would think that since GNMT1 interacts with many lncRNAs, some of them probably in a tissue-specific manner, that they collectively orchestrate DNA methylation. So I just want to emphasize that DACOR1 is not the only lncRNA that interacts with DNMT1. It's not the only lncRNA that is likely to affect DNA methylation. So I think there must be some type of synergistic effects of these lncRNAs in different tissue types. So it's possible that DACOR1 is not expressed in other tissues because it has a tissue-specific function in the colon.
Thank you, Dr. Khalil. Dr. Shen, did you investigate any other types of modifications like ubiquitination or acetylation to P16 in cancer?
No, we haven't done those other type of modification except what has been known based on the literature. So what has been shown in various cell line systems, the P16 methylation is clearly associated with this K9 dimethylation and K27 trimethylation. And also, what we found interestingly is the active markers don't really seem to show dramatic differences. So that's what we have so far, the data, but we haven't done the other kind of modification analysis.
Thank you. Now we have time for just one more question, and it's to both of you. So let's start with Dr. Shen. Can you recommend the best steps for identifying and approaching potential collaborators who may not be in your primary field, for instance, epigenetics?
That's really, really important, actually, for our research to collaborate with multidiscipline area. And as a matter of fact, we were fortunate to collaborate with people actually are expert in the proteinomic studies. So this combination of expertise allow us really to understand further how the epigenetic regulation really plays a role in the cancer biology and how this DNA modification really down to the road mediate the protein function and how this affect the tumorigenesis. I think it is really important to have established collaboration with different areas, not necessarily just in the epigenetic field.
Thank you, Dr. Shen. Dr. Khalil, do you have anything to add?
I completely agree. I think collaborations in this day and age are absolutely essential for good science. We could not have done these studies without important collaborations with colon cancer experts and other experts that not only provided some of the cell lines and reagents, but also provided insights into various aspects, whether they are clinical or translational aspects. So I highly encourage everyone to seek good collaborations that will complement your areas of expertise.
Thank you, Dr. Khalil. That's all we have for today. If you have any further questions, please consider reaching out to the speakers directly. Their email addresses are displayed on the screen. As a reminder, the webinar will be archived on the Scientist website, and you'll receive an email notifying you when the on-demand webinar is available. I'd like to thank everyone who took the time to join us today and for sharing your questions and comments. On behalf of the Scientist, I'd also like to thank our speakers, Dr. Khalil and Dr. Shen, as well as our webinar sponsors, Rockland Immunochemicals, Swift Biosciences, and Enzo Life Sciences. Thank you, everyone, and goodbye.